IEF tampers with the cytoskeleton of the cancer cell and hampers its ability to move. Shown here is a cancer cell with the green visualizing the actin cytoskeleton and the red imaging the nucleus. As you can see, when there is no induced electric field, the actin is able to polymerize to form spokes or these finger-like projections called filopodia that enable the cell to crawl. On the right, observe what happens when we apply our IEF. The actin cytoskeleton is rendered diffuse and unable to form those finger-like projections needed to move. Cell migration requires a lot of energy, and cancer cells use oxidative phosphorylation in their mitochondria, or energy factories, to generate the energy currency that is ATP molecules they need to move. We have shown that our IEF treatment suppresses the activity of a key enzyme called succinate dehydrogenase, or SDH, in their mitochondria that these cells need to convert glucose to this much needed energy currency, or ATP molecules. The runner can thus no longer run now that their knees have been taken out. We are now finding an unexpectedly pleasant surprise in our in vivo animal model results. We have already seen that IEF treatment of mice bearing triple-negative breast tumors not only have their primary tumor growth slowed, but their metastases to the lungs significantly reduced. In the tissues surrounding the primary tumor, as shown in the schematic, application of IEFs causes a favorable immune response in the mice. We find that the presence of anti-tumor immune cells, such as M1-like macrophages, CD8 activated T cells, and monocytic myeloid-derived suppressor cells, abbreviated as MDSCs, are all increased or maintained, while the pro-tumor cells, such as M2-like macrophages and granulocytic MDSCs, are reduced. We find similar immune response in the lungs of these mice treated with IEFs, which supports the observation of fewer to no pulmonary metastases from these triple-negative mammary tumors. Thus, all our data to date show in a compelling way that IEF therapy, either as a standalone treatment or in conjunction with chemotherapy, can effectively treat metastatic TNBC.